that we cure one disease and trigger another one, which was a fatal one. So, and it was the decision from FDA and EMA to start or to open the window for recombinant biopharmaceuticals. Problem with this? Okay, but I could say that this is not the really starting point. The starting point of the protein pharmaceutical, look at this, it's 1893, when Emil Bering analyzed the pores and he got the antibody. So this was the real beginning, but of course it was very primitive in terms of there were no recombinant behind. And look at this now. So this is a really nice ditch. It's a Californian ditch, and just name it. How many companies are there dealing with modern biologicals, production of biologicals and biosimilars? Look at this here. So it is a real mess of all kinds of companies, which means that we are really in the heart of the production, which is in the California. Can I move forward? Okay. Back. So what is a biotechnology actually? It is a composition of different subparts of different knowledges or different areas of research and production. So by using microbiology, bioinformatics, cell biology, in combination with genetic engineering, we are able to produce pharmaceuticals, we are able to produce some enzymes for food industry. There is very important diagnostic and molecular therapy, forensic science, for example. Now you can't say, no, I was not there, I promise you I was not there during the crime. No, because one single cell is enough to detect your presence at the site of the crime. So, I mean, look at this, we are dealing with a lot of different areas. It's not only regarding biopharmaceuticals, but nevertheless, I mean, this is one of the most important parts. So, next, sorry, I have a little problem with it. The biotechnology, pharmaceutical biotechnology, deals with products and services, predominantly for the diagnosis, therapy, and drug delivery as well. It's very important to know that biological medicines could be divided into two basic parts. One is a modern biological production, and the other one is the traditional. And I'm not going to talk about the traditional biological medicines, but predominantly we are talking about recombinant proteins like hormones, enzymes, peptides, cytokines, recombinant monoclonal antibodies, very important part, gene proteins, and synthetic peptides. So let me start with the trends and perspectives. We're going to go through, this will be an overview about what could we expect, what we have in our hands at the time being, and what is our future. So we're talking about first generation of biopharmaceuticals, the second generation of biopharmaceuticals, then we have novel technologies for research and development, and very important is advanced drug delivery. For example, you all know that to cure some cancer diseases that we still use a very old-fashioned drug, doxorubicin, which is a very toxic one. Why not to include doxorubicin together with the antibodies, monoclonal antibodies, in order to deliver the drug directly to the surface of the cancer cell? For example, I'm going to show this later on, biosimilars, gene therapy, personalized drug therapy. So, okay, what is the third generation of biopharmaceuticals? Actually, we are using them for the replacement therapy. What is the replacement therapy? In terms of diabetes type 2 or type 1, we have to introduce into our body insulin, or for example, during different kind of anemias. Anemic status of the patient, we have to inject erythropoietins and so on. So this is the replacement therapy. And antagonist therapy means that we could remove the protein which is the cause of the disease. Like, for example, TNF-alpha. Tumor necrosis factor alpha is a pro-inflammatory cytokine which is guilty in parentheses for a chronic inflammatory disease. So if we are able to remove by monoclonal antibody this kind of molecule, then we could cure or at least relieve the symptoms of the disease. Now look at this. It's a list. It's not a complete one. I mean, I just put some names here, but it's a list of third-generation biopharmaceuticals. We are dealing with more than, I don't know, 70, 80 different molecules like cytokines, hormones, some factors, antibodies. This is far from the optimal. There are many new. Every second 2 months there is one new monoclonal antibodies, and in this afternoon the workshop is going to be dedicated to these antibodies and so on. So you could see a really nice list. Now, what about the market share? This is the global market, but it is in year 2014, which means that at the moment the situation is even more balanced to the biologicals. These are blockbusters, first 10 global. I'm not going to make any advertisement, but just in order to show you that the first 6 blockbusters belong to biologicals, to biopharmaceutical products. Then we have 2 synthetic and again 1. At the moment, in year 2018-19, the situation is a little bit different, but still, I mean, it's even more in favor to biologicals. And look at this, 9 billion, 7 billion, you can imagine the numbers. So this is a real very important part of the modern pharmacy. What are the limitations? Why we are starting with the 4th generation of biologicals? Simply because at that time, or even now, because we have them on the market, quite a lot of them, actually it means that these are biologicals that are produced by different kind of cells, and that we introduce a gene segment coding for, let's say, insulin, protein, whatever, without influence on the structure or on the post-translation modification. So these are the proteins that are counterpart, the recombinant counterparts of our own proteins, for example. And you know that proteins are far from the optimal drugs. What is the optimal medicine? Optimal medicine is the component or the final pharmaceutical form, like a tablet or capsule, that you have in your hand or in your pockets, without being, let's say, influenced on their stability. For example, now I'm pretty sure that you have some kind of acetylsalicylic acid forms of paracetamol in your, I don't know, bags. And did you ask yourself when you enter to this hall, oh, what's the temperature here? Is it stable enough for my aspirin in my pocket? Of course not. So this is the optimal drug, optimal medicine. However, within the umbrella of proteins, this is not the case. I mean, it's very important to know that proteins are far from the optimal. So that's why in terms of solubility, stability, half-life, potency, and immunogenicity, that's why we introduced the 2nd generation of biopharmaceuticals. But before, I'm going just to show you that the production, purification, and characterization of biologicals is a very complex process, and we have to have a lot of knowledge. And look at this. For example, this is the characterization of the 3D structure representation sites. Look at this toolbox of different analytical techniques you have to have in your lab in order to fulfill all the criteria in terms of characterization of the molecule. If you compare this with the synthetic drug, then you could see that this is a real, very complex toolbox. What is also very important is the stability, because proteins are prone to dimerization, aggregation, oxidation, and so on. So stability is also very important. And this is a take-home message for all of you. Immunogenicity. What is immunogenicity? Immunogenicity is a potential nightmare in parentheses if the foreign protein is inserted into our body. But when I'm talking about the proteins, recombinant proteins, you could say, come on, the amino acid structure is the same. Like if we compare with our endogenous insulins, for example, it's true. But for proteins, only one structure is the native structure. We have, the more complex the protein is, the more different kind of structures are potentially present. So only one is a native one. And if the structure is not the same, despite the fact the amino acid sequence is almost the same, or it is the same, it's identical, then this molecule is going to be recognized by our immune system as a foreign protein. And our antigen-presenting cells are going to present this protein for the immune system in order to create antibodies. And this is what we do not want to have. So simple, this is very, very important point. If you compare this with the small chemical entities like morphine, is morphine immunogenic? No. Is a heroin immunogenic? No. If it would be, then we will have no addiction patient or addictive patient. So this is very important to know. If we inject the protein, which is not the really, the structure is not the same like our own, then our body is going to recognize this as a protein and is going to produce anti-drug antibodies, which will completely destroy the activity of the protein. So this is important to know in order to get quality, efficacy, and safety according to the guidelines. And that's why within the European Union we have one single system, and this is centralized system for the registration of biologicals. If the company, if the sponsor would like to have the marketing authorization for generic small chemical entities, no problem. Could be or decentralized procedure or national one or centralized. But for biologicals, only centralized procedure is acceptable, which means that the marketing authorization holder has to apply for registration in EMA in London, and now due to the mess and Brexit and all this kind of funny stories, now the EMA is relocated to Amsterdam. Sorry. Shall I go back? How? You see the problem? Okay. Yes. So just to compare the complexity of the biologicals, guys. So this is an acetyl salicylic acid size. You see 21 atoms, human growth hormone, and a really complex monoclonal antibodies. You could compare this with, in terms of of the structure and the complexity, bike, car, and the jet. I do this. Let me go further on. So just going back to the immunogenicity, what kind of factors might trigger immunogenic reaction? Sequence variation, glycan variation, host cell, contaminants, handling, and storage. For biologicals, it's important to have a cold chain system from the producer to the final consumer, which is patient. Now, let me focus our attention on the safety. What is more important for the biologicals and for all drugs? Let's say this is the general rule: safety or efficacy. 1890. Well, safety, efficacy, it was balanced. Let me convince you by this one. Are you familiar with this advertisement? In 1890, it was an advertisement in which is 40 Stone Street, New York. It was in subsidiary of the biopharmaceutical company, and they introduced aspirin and heroin. Actually, at that time, chemists were very smart, and you know that from salicylic acid, they tried to reduce the side effects, so they acetylated salicylic acid in order to get acetyl salicylic acid, and they named it aspirin. And by the same procedure, they invented heroin. So the name heroin came from the pharmaceutical company, not somewhere under the bridge or under the table. And very soon, look at this advertisement. The chip is specific for relief of cold. So please buy heroin hydrochloric. It's perfect drug. You will never have any cold. You will feel nice and so on and so on. Very soon at that time, it was realized that the side effects due to the addiction, it is so strong that heroin was withdrawn from the market, from the safety point of view. But from the efficacy point of view, heroin is excellent antitussive, believe me. So you see, it's very important to know that the efficacy is more important than the safety and the efficacy. Okay, let me go further on. Second generation of biologicals, we have a lot of different kind of like insulins, erythropoietins, interference, etc. So I will go to skip this because Marco talked too much that he spent all my time. Where is he? Over there. So just to say about this second generation, we could have a protein-protein fusions. We could change the amino acid composition by introduce a new triplet of the nucleotides in order to change the amino acid composition. We could develop a new drug delivery systems like liposome, nanoparticles, microparticles, and very important is post-production modification, for example, pegylation. There is one case, one example. This is non-pegylated interferon alpha. You know that what is the pegylation to introduce the polyethylene glycolic chain connected to the basic protein in order to improve pharmacokinetic and pharmacodynamic properties. So look at this, for example, every second day patient is forced to use an injection or whatever, so that means parenteral use of the administration by pegylated interferon. This is the simple pegylation, and this is the pegylation with the 3 different chains of polyethylene glycol. We could obtain a very stable concentration over time, so one bolus is enough for the patient, so it's very user-friendly. And this is what I would like just to mention briefly about the most important subgroup of the all biologicals as the group of monoclonals, and this was the topic this afternoon. So monoclonals which could be itself a drug or delivery system, and I'm going to skip this because we are short in time. And now let me say a few words about the biosimilars. What are biosimilars? So this is actually the situation when the innovative drug, the patent is expired. What then? You know what is the definition of generics, and I would like to convince you that within the biologicals we have no generic drugs. Why? Because when this is the case of the small synthetic molecule, when the patent is expired, then we could register the generic drug. If there is the same structure, the same dose, the same pharmaceutical form, and this is so-called with the bioequivalence, we could get the generic, which is not the case of the biologicals. Why? Because each cell could produce the structurally different component, and if it's structurally different, that is not then, it's not the component cannot fulfill the criteria for generic. So this is the first producer. You can see this very complex, and this is the different producer. It's perhaps the same genetic sequence, different vector, different process, different downstream processing, formulation. So there are no two identical biologicals, predominantly within the subgroup of monoclonal antibodies. So we have not identical but very similar product, so therefore biogenerics cannot exist. Within the biologicals, we have innovative and biosimilar. That's why EMA introduced several guidelines in order to facilitate the production of biosimilars. Biosimilars are the same biologicals, but are a little bit cheaper, so this simply means that we could see here the biotech drugs and their biogenetic status, and this is the procedure for the assessment of some similarity, marketing, authorization. So even for biosimilar, it's necessary to introduce clinical studies. This is very important to know. For generics, we have no clinical studies. For biosimilars, yes, there are observed clinical studies compared to clinical studies. About gene therapy, I'm not going to tell you a lot. It was a real disappointment, but now we are quite happy in the last 2 or 3 years. But let me compare the gene therapy with the mobile phone. This was the first mobile phone. It was a mobile and it was a phone. You know, this is a vehicle which was used during the beginning of Second World War, even before, when German soldiers, he was a driver and he was an expert, a man who was responsible to communicate with other cars. So this was a mobile phone. And now what we have here in our hands, our field. So gene therapy, we are somewhere here. Could you imagine what kind of progress we have to do in the next 20 or even more years? So yeah, this is about gene therapy. Very simply, we could introduce the genes. So what is the gene therapy? It is the part of biologicals, but we are not dealing with proteins, but simply with the genes that are introduced into the cells by different way, by viral or non-viral gene transfer. So now let me conclude all about this, that modern biologicals are really produced by recombinant technology, and we are dealing with large, very sensitive molecules. So not many companies in the world are able to produce very, let's say, quality products like biologicals or biosimilars. We are dealing with non-comparable biologics coming from some obscure companies, and within the European Union we are happy that they are not allowed to penetrate, to enter into the market. Then at the time being we have a lot of products or peptides, proteins or peptides that are market, and that we have to know that immunogenicity is one of the most specific and severe adverse effects that might occur or during the transportation or during the treatment, and that we have to put a lot of attention for the quality proteins. And the very last slide, despite the fact that we are in beautiful town Poreč, I'm not put something like this one, but allow me to make a little bit of advertisement to my own country, Slovenia. This is the only country with the word love. So I wish to thank you for your attention, and I'm open for discussion perhaps during the class. Thank you.
If there are any questions, do we have any more questions for Professor Poreč? If not, we can just take questions later for panel discussion, if this is okay too. I would thank Professor Poreč for amazing lecture. We've been crossed the time limit. However, we are now moving to the next lecturer. So it is a great pleasure to announce our next speaker. So this is Robert Likic, who currently works as an associate professor at the Zagreb Medical School and as a consultant internist at the University Hospital Zagreb. The main focus of Robert's lecture will revolve around the development of novel biological compounds, differences between originator and biosimilar molecules, CAR-T-cell treatments, as well as gene editing technologies. So let's hear next 20 minutes lecture, and Professor Likic, you have your floor.
Okay, thank you, Marco. For the upcoming speakers and for myself after this morning, there was a slight problem with the pointer, hence I'll be closer to the...
No, it's working, it's working.
Oh, it has been fixed?
Yeah.
Nevertheless, I will keep moving, and let's hope that it won't have any problems. So first, a disclaimer. And then secondly, that. Yes, so in my talk, I will be covering 4 fields in treatment that I think will be of strategic importance for you in the time to come. As Marco told you, I'm a professor of internal medicine working for the past 15 years at the Unit for Clinical Pharmacology. I'm also an editor at the Journal of Clinical Pharmacology. So I will be focusing on biosimilars, immunotherapy, especially checkpoint inhibitors, CAR, and then the application of artificial intelligence and machine learning to the field of therapy. I think you, as predominantly pharmacy students currently, would do pretty well to focus on one of these 4 strategic fields in your future careers. As you can see here, this is actually from Bloomberg, which is one of the business intelligence companies, leading business intelligence companies in the world. And this graph shows you how the pharmaceutical industry worldwide is among the companies who are investing the largest amounts of money into research and development. Here, among these 20 worldwide companies, we have 8 pharmaceutical companies investing largest amounts of money into the research and development fields. Hence, this field or this industry is extremely competitive. And as you know, development of new compounds is prohibitively expensive. Certainly I, with my little company, my garage, could not put a new compound on the market. Even in the times of Yugoslavia, when we had Pliva as the flagship pharmaceutical company in that big country with 20 million people, even Pliva couldn't put azithromycin, that was our national antibiotic. Even they could not afford to put it to market. They had to establish a partnership with Pfizer and then sell marketing authorization for Western Europe and Northern America to Pfizer for the funds to assess and evaluate the compound and eventually put it to market. Hence, it's prohibitively expensive. One of the things that pharmaceutical industry is looking for when they're trying to develop new drugs is actually how to have greatest profits. And as my previous presenter told you, biologicals are extremely profitable compounds. Predominantly since these drugs are effective. They solve a need in the treatment of patients that cannot be addressed with other therapies. Hence, worldwide the market for biological drugs is on the rise, and this is like linear. It's going to be exponential. And you see here it has passed the number of $200 billion US 2 years ago. So it's a very, very lucrative field for research and for development of new compounds. So much so that today, anybody who is somebody in the pharmaceutical industry, all the big players are investing and developing biosimilars. As you've already heard, biosimilars are to biologicals what generics are to original chemical molecules. They are not identical because our current technology at this time, in this place, cannot guarantee that they are 100% same. Okay? One could go even that far and say that today's originator biological is biosimilar to its own molecule from a year ago, to a different batch. Because since they are produced on cells, we cannot be 100% sure that every batch is totally identical. Hence there is the term biosimilar, or actually what you also find in the literature is being used is the term indistinguishable. So we could say biosimilars, as per our current technology, are indistinguishable from the original biological molecule. The reason is that these molecules are extremely complex. You've seen examples earlier, but just to reiterate and stress the point once more. Infliximab is like a Titanic ship. It's a huge molecule, a lot of immunogenicity there. Then filgrastim, another biological molecule, is like a boat. And to these two molecules, acetylsalicylic acid, aspirin, is like a screw. Okay? Huge size difference, huge potential for immunogenicity. So on the former slide, I'll just bring it back. So you see that the market in the European Union in billions of euros for biosimilars is huge. And with the patent cliff, all of these molecules, all these drugs are blockbusters. So with the time and patent cliff, we have appearance of a number of biosimilar molecules on the market. Since they are cheaper, they will take greater portions of market of the originator drugs, and you as future pharmacists will be probably very likely faced with the duty to counsel either patients or other healthcare professionals on the aspects of these treatments. Are they switchable? Are they identical? Do they share the same route of application? What are the dangers? I mean, are the dangers of biosimilar compounds similar to the dangers of originated compounds? Yes, they are. Are they greater? No, they are not. Can they be switched? Of course, there's a number of studies done so far that have not found any toward effect coming from switching of these molecules. And then this is potential for biosimilar molecules appearing on the market in the time to come. As you can see, a number of very active compounds among some of the most effective biologicals, checkpoint inhibitors like pembrolizumab, ipilimumab, pertuzumab for mammary cancer, and then treatments for multiple sclerosis. All of these drugs are biologicals, and yes, they are effective. It's really impressive to see multiple sclerosis patients who had their debilitating disease suddenly stabilized owing to these treatments that have recently become available. What are the trends in Europe? We in Croatia have a joke. The trends become poorer the more to the south you go. So we are rather conservative here, and in this country, or in this region, I would exempt Slovenia from the region. They are more Western and more advanced than we are. We tend to be conservative, and reforms here are slow. So biosimilar penetration in this country is not that successful. It's not bad, and depending on the time of entry, it's even better than in some other countries, but in general, it's not so great like in Scandinavia, where you have like Norway, who were at the vanguard of substitutability and switching between biosimilars. You perhaps have heard of the Norwich study where Norwegians switched their rheumatological patients from originator originator infliximab to biosimilar infliximab, published about that in the leading journals. Nothing happened. So their studies were actually pivotal in promoting the switch and actually savings, since if you achieve savings by switching or promoting biosimilar prescribing, then you save funds, and you can use these funds for other needs in the healthcare sector to afford novel treatments, of which you will hear later on. And here's the time to tell you something about the new treatments, about the actually one of the revolutionizing treatments in oncology. This rather busy slide shows you the concept behind checkpoint inhibition. You all probably know that there are malignant tumors that are poorly responsive to treatment. The primary example I could name is pancreatic cancer, which is actually 100% deadly, and it very seldom probably see drugs that have been allowed. Okay, so we now have, for the first time in history, I would say, treatments that are promising in this regard. I wouldn't say that they solve everything, but I would say that they are promising. Treatments actually that target the PD-L1 and PD-1 molecules. PD-1 stands for programmed death receptor. PD, or actually one could say receptor for apoptosis, is a receptor that's found on immune cells. And it's actually important when it comes to downregulation of the immune response. And typically what gets bound to this PD-1 receptor is the PD-1 ligand, a molecule that's again secreted during the immune reaction, and it suppresses the immune reaction so as to preserve its own tissue. However, hard-to-treat tumors have actually adopted a strategy to secrete PD-L1 ligands. Okay, so they behave like our own tissues, our own cells, and yet suppress the immune system from attacking them, causing the immune cells to become zombie-like, dormant. They go to sleep, and they do nothing. And then the tumor spreads, and eventually kills the patient. Despite our best efforts to control the disease, eradicate it, try to treat it with methotrexate, doxorubicin, the patient is dead. However, so now we've developed monoclonal antibodies that actually bind either to the PD-L1 molecule or to the PD-1 receptor, like nivolumab and pembrolizumab, monoclonal antibodies, bind and block the PD-1 receptor. Hence, the ligand cannot bind to it, and then atezolizumab and durvalumab bind to PD-L1 molecule. So it itself cannot bind to the receptor. What you have stemming from this is blockade in here. So you have a block in the downregulation of the immune response. And that's like switching the immune system into high gear. So instead of it being parked, like the tumor intense, it's like, okay, go into gear number 6, overdrive. And we have then a tremendously increased immune response towards tumor, usually towards everything else. This is sometimes also referred like cytokine storm, but in this instance, it tends to be directed against the tumor. So there are actually 3 types of molecules that are involved in this strategy that is gaining traction every day, and I'm convinced inventors of these drugs will probably receive a Nobel Prize. So you have like nivolumab and pembrolizumab, that will be, I think, Opdivo and Keytruda. It's actually 2 drugs with the same target. I would say they are switchable. I can hear it here from me. I would say they are switchable. One was developed in Japan, another one in the United States. And when I was at the World UFARS Congress in Kyoto, Japan, I think last year, then nivolumab was, all the Congress was in the flags of nivolumab. They are really in awe of their achievement. I mean, we all should be. So this is one strategy, you block PD-1. Then you have PD-L1 blocking agents, atezolizumab and durvalumab. So that's another insert block. And then The third strategy is this here molecule, CTLA4 inhibitors. One of them is ipilimumab, that would be Yervoy. Current studies suggest that in hard-to-treat tumors such as melanoma, pulmonary cancers, kidney cancers, urinary bladder cancers, this is the optimal strategy. My colleagues at the Department for Pulmonary Oncology and at the Department for Urinary Genital Tumors have had or do have patients that have exceedingly well responded to this combination therapy, like stage 4 malignant disease with metastases in the lungs, with metastases in the liver, after operation and combination of nivolumab, ipilimumab, and 2 years later, the patient is still alive, okay, taking care of his farm animals, like not chickens, but pigs and cows, so working full time. So the results are currently pretty amazing. To show you that, or to stress that a bit more, these treatments are slowly becoming first-line therapies. They are prohibitively expensive, and that's only what is blocking the move to first-line therapy. So it's still being reserved for tyrosine kinase inhibitors, but for advanced diseases with poor or immediate or intermediate risk, you have it. Okay, here, even in Croatia, these treatments save lives. Will they save health budget from bankruptcy? They will not. They will cause the budgetary bankruptcy for healthcare in Croatia and elsewhere, Sweden, UK, anywhere, because these treatments are expensive, but they are effective, and I would say they are safe. To demonstrate it a bit more, here you have a metastasis in the lung. Look how over time it gets smaller, and this is actually a scar tissue. It's inactive. So quite effective therapies. So what can happen with them? Or what can they cause with regards to side effects? Because a completely different profile of side effects in contrast to the standard therapies. So you have like immune-related side effects that can manifest, such as eye inflammation, dry mouth, various forms of rash, myocarditis, hepatitis, thyroid disease like thyroiditis, colitis, pancreatitis, even sometimes myopathy. Okay, a different set of side effects, and sometimes I will highlight it in more detail on my workshop. Sometimes these side effects do not mean that the treatment is poorly tolerated. Exactly the opposite. They mean that the treatment is extremely effective. Okay, but more on that for those who come to my workshop. Also, the side effect profile of checkpoint inhibition is different with regards to time as well. So the first thing that will develop, if the drug is poorly tolerated, is rash with pruritus or skin itch, then diarrhea, then patients can have hypophysitis and liver toxicity. All of this is manageable. You can decrease the dose, you can give corticosteroids to stop the treatment. But usually side effect profile is such that it does not cause grade 4 side effects, meaning you need to stop the treatment. Moving on to the third field that I want to showcase to you, and this is probably, I wouldn't say the most important slide you will see today, but one of the more important slides that you will see today. It can decide actually how good you fare in your future careers. So this slide shows you a statistical power. So whatever you do, it actually makes sense to be aware of this statistical law. To put it into lay's terms, if you're measuring an effect, so effect size, of an intervention or of a drug, the greater the effect of the drug that you are measuring, like here, this means that you will require fewer patients or test subjects to prove that your effect is not coincidental, but that it is due to your intervention or to your drug. Okay? Meaning, the more effective your drug is, the sooner you will prove that the effect is due to the drug, the fewer patients you will require, and the whole process will be significantly cheaper. To give an example for this, a few years ago, I was reading a paper in the New England Journal of Medicine. This paper was published from the Penn State University, and it described a trial enrolling patients with treatment-refractory chronic hepatic leukemia. Essentially, these 3 patients have undergone all lines of treatments available for chronic lymphatic leukemia, and they failed all the treatment plans. And that's when in the United States, your doctor would say something you don't want to hear. Right? So that's when they would say, get your affairs in order. Okay? This means, you're about to die. So prepare yourself and your family for it. So you don't want to hear that over there. And what happened then was that they had a clinical trial testing a very novel treatment strategy. So they would take the T helper cells from the patients, use a viral vector, in that case it was an HIV, where the genome was replaced by a genetic construct, like an amalgamation of genes specific for pan-B lymphocyte receptor, then another gene for boosting the immune response, and the third gene for the co-receptor for the B lymphocyte receptor. So they use the B lymphocytes of the patients and mix them with the HIVs containing the genetic construct. What they ended up with was transgenic patients' B lymphocytes. And then every patient received their own, now modified and improved transgenic T lymphocytes. And consumed was a flu-like disease in all 3 of them. And 2 weeks later, when they re-evaluated the patients, not a single tumor mass or not a single enlarged lymph node was to be found. It was amazing. For all purposes, all 3 patients were cured instead of dying. What they also found was that no circulating B lymphocytes could be detected in the bloodstream of the patients. But this is compatible with life. So for 6 months, these patients, the 3 of them, would receive IV immunoglobulins. And hence, this was the first trial that only 3 patients got published in the New England Journal of Medicine. And this is the beginning of the heart care. Huge effect, they would have died. The effect is actually somewhere there over the swimming pool. And they required only 3 patients to prove it. And they ushered a new era in the treatment of hematological malignancies. Okay? Another Nobel Prize will go for this. So this was brought to us from the Penn State University, and the patent was then sold to the Novartis. A few years ago, when Lipids was traveling abroad, I noticed on one airport at the kiosk there was a huge Forbes journal headline with the chief executive officer of Novartis claiming we will cure cancer. Big claim. But actually, yes, they will, only not all types of cancer. They will cure, likely, all types of cancer stemming from B B lymphocytes. Okay? And this is why, so again, CAR T-cell therapy is actually, you take the T lymphocytes from the patient, then you use viral vector, and then every company has its own. In the instance of Novartis, which purchased patents from the Penn State University, they are using HIV. Other companies are using different vectors. So they use a viral vector to insert genes into the T cells, thus changing these T cells to express chimeric antigen receptor, and they multiply them. All of this is done, by the way, outside of the hospital. In this model, all of this is done outside of the hospital, and usually takes the company 4 to 6 weeks to treat the cells, and then they ship them back to the hospital where they get infused to the patient. So you have a critical window of treatment failure here. If your patient is severely ill, they might die in the period that the transgenic T cells are being multiplied. And then they are returned to the patient, and then they fight against the disease, and typically get rid of the disease. So it's a solution, a perfect one, for lymphatic leukemia, chronic one, and a good portion, a greater portion of lymphomas, non-Hodgkin and Hodgkin lymphomas, as well as plasmacytomas, all of which are dreadful diseases, and in all, right? This treatment or this innovation will get a Nobel Prize. People who came up with a strategy. And there are two compounds. Now we are well advanced in this. And there are two compounds that are registered that will be used in the European Union soon. One is Kymriah, so that's the original strategy about which I told you, and the second one is Yescarta. One is Novartis, Kymriah, and Yescarta is Gilead. Very effective, especially in children, where the first use will take place. This slide actually shows you what happens when this treatment fails. This is again from I think New England, Nature Reviews, but what can happen, what can cause problems is that not enough T cells can be harvested, then they cannot be multiplied, and then this is a big problem, that tumor cells lose the pan-B lymphocyte receptor, CD19 or CD22. Since these monoclonal antibodies target these receptors. And then you have side effects of cytokine storm. Currently, today, they use tocilizumab, another monoclonal antibody against interleukin-16 to control the immune response to treat it, so as not to kill the patient, to only kill the leukemia. And then the final and biggest problem is this. As I said, this treatment currently works only for B lymphocytes. Forget solid tumors, some lymphoma subtypes, and malignant diseases of the central nervous system, like glioblastoma. Glioblastoma is the most malignant, radio-resistant, chemo-resistant tumor of the central nervous system. Those who develop glioblastoma are destined to die within 6 to 12 months. Nothing works. I, in my crystal ball, see perhaps rabies virus working for glioblastoma. It's a virus that's aggressively neurotropic, and if you could change the genome of the rabies virus, you could perhaps use it to transport an anti-tumor load to the central nervous system and kill glioblastoma cells there. So that might be a very, very distant future. It's no wonder that HIV was used as a vector, since it causes AIDS and was the most heavily studied virus of them all throughout history. And then finally, coming to the fourth strategic area that I want to draw your attention to. This is the machine learning and artificial intelligence field, where today, even I on my crappy computer can run machine learning algorithms and can predict factors that would cause a traveler on the Titanic ship to die during that catastrophe. So you actually can train your model and then use your trained model of machine learning, let's say in Python, to predict the outcomes for a new set of data. In this instance that I'm showing you, this approach is being used for glioblastoma. So this is a very malignant tumor, and they use POC regression to predict survival of patients in this disease. Well, let's give them on this example, it can also go wrong. This is the example of the Watson supercomputer. It's an AI system developed at IBM who was trained poorly. And this can have disastrous effects. So, if trained poorly, such systems can give wrong predictions. Like use methotrexate to treat pregnant women. Yeah, you all know how that went. But there are multiple trials ongoing all around the world. This is from the Lancet and clinicaltrial.gov. At least 150 trials ongoing of AI systems worldwide aimed at improving treatments and improving survival. And the latest trend is to do genetic analysis of the tumor tissue. Here you've heard of Human Genome Project. This is a system based on Tumor Genome Project. There you have 22,000 genes of various tumor types analyzed, available for all to download worldwide and analyze further. So you can do it on your own, or you can have professionals do it. This is a sample of the finding from Foundation 1, from Roche. You send them a tumor sample and they analyze it for some 400 gene mutations. And then they use cross-population and cross-match to find clinical trials for drugs that are available in other indications or mutations that have been detected in this test. And then you can try and prescribe this treatment for a tumor that your patient has, who might otherwise would not receive the treatment that they would require. So I hope this will not become the reality very soon, like that we all or the patients consult Siri and not pharmacists or health professionals, but this just shows you that AI and machine learning is one of the bigger trends that we need to be aware of. And this I would like to conclude my talk. Thank you.
Thank you very much, Professor. Thanks for an amazing lecture. And do we have any questions, perhaps? If not, we can then leave questions for panel discussion, and we can now move to our third lecture. So it is a great pleasure to announce our next speaker, and that speaker is Norma Kessler, who is currently working as a postdoctoral researcher at the Faculty of Pharmacy and Biochemistry, University of Zagreb. During his lecture, you will have an opportunity to learn about protein lipozylation and its importance in health and disease, and see how glycoengineering could have enormous importance in the development of the next generation biopharmaceuticals. So let's hear next 20-minute speech from Professor Kessler. So you have the floor.
Thank you, Marco, for the introduction. So we are a bit out of time, so I will try to be quick. So my lecture will have two parts. First, I will tell you something about glycobiology, And in the next part, I will tell you about the importance of being glycosylated in pharmaceutical sciences. So first, introduction to glycobiology. So I would like to emphasize the difference between two terms which are often mixed up. It's glycosylation and glycation. So it's definitely not the same thing. So glycation is part of physiological process. It's consequence of diabetes mellitus. It's when some simple sugar structures, most often glucose, is attached to proteins, which impairs their function. So it's pathological. And glycosylation is definitely not pathological, so it's complex, highly specific, and strictly regulated co- and post-translational process. So co-translational means that these complex sugar structures are attached to proteins while they're synthesized in endoplasmatic reticulum. And post-translational means that after the protein is synthesized, this glycan part is further modified in endoplasmatic reticulum and Golgi apparatus. So it's mediated by hundreds of enzymes and it has numerous important physiological roles. So what are some of the functions of glycans? So they have important structural role. They build up cell walls and extracellular matrix. If they are bound to proteins, they modify their functions, stability and solubility. And by interactions with proteins, they are directing trafficking of glycoconjugates and they are mediating and modulating cell adhesion and cell signaling. We can find glycans in all living organisms, and multicellular life without glycans is not possible. So if you have a major defect in glycosylation, it's practically embryologically lethal. In humans, it's estimated that about 70% of all proteins are glycosylated. So glycans can be bound to proteins or lipids. So they can be bound to proteins via N linkage or O linkage. N linkage is with asparaginase protein, and O linkage is with serine protein. And if they're bound to lipids, they're called glycolipids. So why are these glycans so interesting? Because they have a huge potential diagnostic and prognostic value, so they change many diseases. And mostly inflammatory diseases such as rheumatoid arthritis, inflammatory bowel disease, acute pancreatitis, sepsis, cancer, and so on. And what's the problem with glycosylation? The main problem is that glycoanalytics is really hard. Because you can have multiple glycosylation sites on the same protein, and each of these sites can have numerous different types of glycan attached, and each of these glycan has a large structural complexity because of its monosaccharide composition, anomeric state, linkage of the subunits, branching, and linkage to the peptide part. Just to give you an example, so if you have a pentamer which is composed of 5 monomers, so if you want to build up a small protein, so oligopeptides from amino acids, from 5 amino acids, you can have 120 different combinations. But if you want to build a small glycans from 5 monomers, so from 5 monosaccharides, you can build more than 2 million combinations. And because of this complexity, most researchers just ignored the glycan part of the glycoprotein and they just concentrated on the protein part. And because of that, the knowledge about the role of glycans has been lagging behind the knowledge about the role of genes and proteins. But for example, this glycoprotein has 40% of its mass discovered by glycans, so you cannot really expect to understand the function of glycoprotein without knowing the function of the glycan part. But thankfully, recently major progress has been made and high throughput analytical techniques for glycan analysis have been developed. So our glyco team from Zagreb, both from university and GENOS, we analyzed glycans in over 1,000 samples from some of the best genotype and phenotype cohorts in the world, but we mostly concentrated on total plasma and IgG glycans. So there's still much knowledge missing. Because of that, Professor Gordon Laus from our team, together with Professor Richard Cummings from Harvard Medical School, last year they started the Human Glycan Project, and the goal is to understand the role of glycans
is both incumbent disease and it's connecting the glycobiologists from all over the world. Okay, now to the second part, the importance of being glycosylated in pharmaceutical sciences. So I will again divide this part in two parts. So first I will talk about the influence of drug glycosylation on interaction between human body and drug, and in the second part, the influence of human glycans, so glycosylation of human proteins on interaction between human body and drug. Okay, so first this first part, influence of drug glycosylation. So most biopharmaceuticals are practically glycoproteins, and they include monoclonal antibodies, erythropoietin, cytokines, growth factors, therapeutic enzymes, and so on. And these glycans can have a profound effect on their stability, activity, and pharmacodynamics and antigenicity, so immunogenicity. I will first tell you about the importance of glycans for stability on example of erythropoietin. So erythropoietin is a glycoprotein and it's secreted by kidneys in response to cellular hypoxia. So if cell lacks oxygen, it will stimulate kidneys to release erythropoietin, and this erythropoietin stimulates red blood cells to red blood cell production. And erythropoietin is highly glycosylated, so 40% of its molecular weight are practically glycans. It has 3 N-glycosylation sites and 1 O-glycosylation site, and if you remove these glycans from the molecule, erythropoietin will have less than 10% in vivo activity compared to glycosylated form. Why? Because it will be faster removed by the kidneys, and if you remove just 1 monosaccharide from this glycan portion, it will be faster removed by liver. So we can see that erythropoietin, glycans and erythropoietin have a great influence on its stability in blood. Because of that, now we have on the market diarypoietin alpha, so it's a re-engineered form of erythropoietin. They made a few amino acid changes in the protein part, and this resulted in creation of 2 new N-glycosylation sites. So the original molecule, this one, has just 3 N-glycosylation sites, and this re-engineered has 5 N-glycosylation sites. And because of these 2 additional glycosylation sites, this diarypoietin alpha has threefold longer serum half-life compared to the traditional erythropoietin, and it requires less frequent injections, which is really convenient for the patients. Okay, now next thing, so I will tell you why glycans are important for activity in pharmacodynamics on example of monoclonal antibodies. So most monoclonal antibodies are immunoglobulin G. So you all know for the function of immunoglobulin G that it binds antigens with this variable FAB domain. But this FC constant domain of IgG has an equally important role because it comes in interaction with FC receptors which are present in many immunological cells. So you can see both of the functions in this small picture here. So with FAB region it binds to pathogens, and with this FC region it binds to FC receptors on immunological cells. And here on this picture you can see many types of these FC receptors which are present on many immunological cells. And this FC region of immunoglobulin G always carries two glycans. So practically all monoclonal antibodies have these 2 glycans in this FC region. And in most cases this glycan looks like this. But if you just add 1 galactose or 1 sialic acid, this monoclonal antibody will no longer be pro-inflammatory, it will be anti-inflammatory because of the change in interaction with these FC receptors. And for example, if you remove this 4-fucose, you will increase binding to FC gamma R3 receptor by about 100-fold and increase antibody-dependent cellular cytotoxicity. So why is this important? This antibody-dependent cellular cytotoxicity, so studies on rituximab and trastuzumab have suggested that ADCC is the key mechanism of action to eliminate cancer cells by monoclonal antibodies. And by lowering this 4-fucosylation, you can increase this interaction between immunological cell and IgG, which is bound to cancer cell, and this increases its efficacy to destroy cancer cell. So because of that, there are currently 3 efficacy-related antibodies in the market, and more than 20 of them are currently being evaluated in clinical trials. So one example of antibody which is already on the market is obinutuzumab. So it's first glycoengineered therapeutic anti-CD20 antibody approved in 2013, and it's first-line treatment for chronic lymphocytic leukemia. And this antibody has only 30% fucose level of a normal antibody, and because of that, it has superior tumor inhibition compared to rituximab. Another 2 which are on market are mogamulizumab and benralizumab, which was improved in 2017. So the first one is for treatment of hematologic cancers, and the second one for treatment of severe eosinophil asthma. Okay, and the last, why glycans are important? Because they have an effect on antigenicity, immunogenicity. So all biopharmaceuticals are practically produced in cell lines, and there are risks arising from non-human glycans that could cause excessively rapid clearance or immunogenic reaction. So, for example, if you produce your biopharmaceutical in an insect cells produce this type of glycans, which is not present in humans, so it will be immunogenic, it will cause immunogenic reaction. In plants, you have this xylose, which is also immunogenic to humans, and in yeast, you have this hypermonazylation, which will cause rapid clearance by kidneys. Okay, so then you might think if we go to mammalian cells, things will be better. But also in mammalian cells, you have this type of glycans which are not present in humans and which are antigenic to humans. So conclusion of this part, so control of glycosylation is of major importance during the development of biopharmaceuticals because they influence, as I told you, on immunogenicity, on activity and stability in blood. And because of that, they are proclaimed as critical quality attribute during the biopharmaceutical development. So glycoengineering has already started, so we need to design and control the type of sugar molecules in a specific region of biopharmaceuticals. But there are still some problems, as I told you. Production in cell lines have problems with foreign glycans. So enzymatic glycan remodeling cannot really help because it's complicated and expensive. And maybe the key will be playing with the genome of the cell lines which produce these biopharmaceuticals. So maybe if we remove genes which produce non-human glycans and put genes which make human glycans, then we can solve this problem of antigenicity. But altogether, glycoengineering has potential to develop the next generation of biopharmaceuticals, but we still need to learn more about glycosylation and how to control it. And now the second part, so influence of human glycosylation on interaction between drug and human body. So you all heard for personalized medicine. So the same drug at the same dose can have a total different effect on different subgroups of people. And the major challenge is how to identify these subgroups of people. One of the answers is pharmacogenomics. So by analyzing genes of these individuals, we can predict which individuals will need this exact dose, lower dose, larger dose, or some totally different drug. But genes cannot explain all the differences, and maybe some answers could be in glycans. So I will show you this on an example of warfarin. So warfarin is one of the most popular anticoagulant drugs. It's used to treat deep vein thrombosis and pulmonary embolism and to prevent stroke. So I'll not go in details in pharmacodynamics and pharmacokinetics, but I will tell you that it has really narrow therapeutic index and large inter-individual therapeutic dose variability, so up to 20 times. Okay, and because of this narrow therapeutic index and large inter-individual therapeutic dose variability, we have a dosage problem because too much or too little can be lethal for patients. If you give them too much, then they can bleed to death. If you give them too little, then you don't have this protective effect against cardiovascular events. And And because of that, the patients have to go to hospital every day, do blood testing to check how this dose is working on them, and this can sometimes last for months. So can personalized medicine help? Okay, pharmacogenomics of course has some answers. So if you test polymorphisms in these two genes, it helps with the prediction of the right dosage, but it's not really effective in extreme dosage cases. So could glycosylation help? Before I go to glycosylation, I will tell you that 97% of warfarin in human blood is bound to plasma proteins, and they're mostly bound to albumin and alpha-1 acidic glycoprotein, and only this unbound fraction of 3% exhibits pharmacological effect. And this alpha-1 acid glycoprotein is heavily glycosylated, and depending on which glycans you have on this AGP, it will influence on interaction between warfarin and AGP. Why? Because these glycans are near the site where warfarin bounds to this AGP. So when the theory is, if you have different AGP glycosylation, which most people have, you will have different affinity of AGP to warfarin, you will have different amount of warfarin unbound fraction, you will have different pharmacologic effect, and patients will need different warfarin doses. So the main question is this clinically relevant? If yes, then this could be the dawn of pharmacoglycomics, and it means that different protein and glycosylation can cause different drug response. And currently we are testing this in our lab. Okay? And then if this works, then pharmacogenomics and pharmacoglycomics together would help to predict the right dose so that patients don't have to go to hospital each day for months. So the conclusion of this part, pharmacogenomics and pharmacoglycomics could together help us to accelerate the adoption of personalized medicine so that the right patient gets the right dose at the right time, and this could improve the quality of patient care and help to contain healthcare costs. Thank you.
Thank you very much for the presentation and great lecture. Do we have some questions for Roma? I see none at the moment. We will then leave those for the panel discussion. Thanks once more. And now we will proceed to the coffee break. But before we get out, I have just a few announcements. So first, coffee break will last until 11:45. No, 11:40. 11:40 will last the coffee break. And further, be aware that you need to check in when you come back. So in order to collect your stamps for evening program, and as well